Welcome to Bio Bits and Bytes. Today we're plunging into the really dynamic world of immune cell interactions. We're zeroing in on the surprisingly multifaceted roles of B lymphocytes.
hmm
We'll be looking at how they're involved in two areas that seem quite different on the surface: adipose tissue inflammation and some really innovative strategies in cancer treatment.
That's right. We've got some fascinating recent findings that really highlight just how adaptable and, well, essential these B cells are in different biological situations.
Absolutely. So we're going to kick things off by examining how B cells behave within adipose tissue, that's our fat tissue,
Yeah.
and how their actions there might link up with metabolic health issues.
Okay.
Then we'll shift gears pretty significantly and investigate the exciting ways B cells are turning up in a new frontier of cancer therapy.
Sounds good.
Now, immunology, as you know, it's got its own language. So as we hit any specific scientific terms, our expert here will give us some crystal clear explanations. We want to make sure everyone can follow along.
Happy to help break things down.
Okay, let's unpack this. First up, adipose tissue inflammation.
Great place to start. So the research we're looking at really highlights B lymphocytes living within adipose tissue, which we often shorten to AT.
AT, got it.
Now, it's important to realize AT isn't just, one uniform blob. It's organized into different depots, mainly visceral adipose tissue and subcutaneous adipose tissue. Should I maybe explain the difference there?
Yes, definitely. Let's build that solid foundation first. What are VAT and SAT?
Certainly. So visceral adipose tissue, or VAT, that's the fat surrounding our internal organs.
Okay, the deep stuff.
Exactly. Think of it like packing material around your stomach, intestines, inside your abdomen. The research mentions specific types in mice, like omental epididymal, which is near the gonads mesenteric, even around the heart, like epicardial and pericardial and perivascular, meaning around blood vessels.
Wow, okay, so this is in a lot of crucial spots.
It is. And then subcutaneous adipose tissue, SAT, that's the fat just beneath our skin, the stuff you can pinch. Then there's also brown adipose tissue, BAT. That one's a bit different, more involved in burning energy, generating heat. And interestingly, B cells pop up there too.
Okay, and B cells themselves, just a quick refresher.
Sure, B lymphocytes or B cells. They're a key part of our adaptive immune system. Most famous, probably, for making antibodies.
Antibodies, those are the targeted responders, right? The proteins that latch on to specific things.
Nicely. Antibodies, also called immunoglobulins, you'll see names like IgG, IgM pop up. They recognize and help neutralize specific threats, which we call antigens.
Okay.
And even within the B cell world, there are different teams, like B1 and B2 cells. They have slightly different jobs.
Right.
We'll also bump into signaling molecules called cytokines. Think of them as messengers. Some, like TNF-alpha, tend to promote inflammation. Others, like IL-10, are more anti-inflammatory. They help calm things down.
Inflammatory and anti-inflammatory messengers. Makes sense.
And then there are chemokines, like CXCL13. These act more like beacons, calling other immune cells to a specific location.
So cytokines are the message. Chemokines are the come here signal.
Kind of, yeah. A good way to think about it. And on the B cell itself, the B cell receptor, or BCR, is its unique tool for recognizing a specific antigen. It's like a lock that only a specific key fits.
Gotcha.
B cells also need other signals, costimulatory molecules, to get fully activated. It's like needing two keys to turn the ignition.
Okay. And quickly, some of the metabolic terms we might hear.
Right. We'll likely talk about insulin resistance, or IR. That's when your body cells don't respond properly to insulin, the hormone that helps manage blood sugar.
Which is linked to things like type 2 diabetes.
Exactly. And metabolic syndrome, or MET-DS, that's a cluster of conditions often occurring together. Things like insulin resistance, high blood pressure, unhealthy cholesterol levels, and often excess abdominal fat, that VAT we mentioned.
Okay.
And finally, you'll hear about high-fat diets, or HFDs. Researchers often use these in animal models to mimic some aspects of human obesity and study those related metabolic changes.
That's a fantastic primer, thanks. So the research seems to really stress that where the fat is located makes a big difference for the B cells found there.
That's a really key takeaway. The studies absolutely show that the number of B cells and the specific types of B cells present can vary a lot depending on the AT location.
So VAT versus SAT is different.
Definitely different. And even within VAT, comparing, say, the omental VAT to other visceral depots in mice, you see distinct B cell populations.
Omental VAT, you mentioned that one specifically. Does it have a special role?
It really seems to. Omental VAT is strategically placed, connected to organs like the spleen, stomach, pancreas, colon. It sort of drapes over other abdominal organs.
Made an apron?
A bit like that, yeah. In mice, it's actually quite small, like a little strip near the stomach. But it plays a big role in moderating inflammation within the abdomen, helps with healing after surgery, promotes new blood vessel growth, and helps limit infection and inflammation. It's sometimes called the policeman of the abdomen,
Wow, policeman of the abdomen. I like that.
and it's a major hub for immune cells. It formed these organized clusters called milky spots.
Milky spots.
Yeah, little white spots visible on the surface. They're packed with immune cells, especially rich in those B1 cells, a more innate-like B cell, and also macrophages. We see this in both humans and mice.
So a real hotspot for B1 cells in the omentum.
Right. And these B1 cells are quite mobile. They can actually travel between the chest and abdominal cavities through the omental VAT, and that movement seems directed largely by that chemokine we mentioned, CXCL13, which is made by macrophages living there.
Fascinating. What about these FALCs you mentioned earlier? Fat-associated lymphoid clusters.
Ah, yes, FALCs. Good question. These are another type of immune cell gathering. They were first found in the fat around the intestines, the mesenteric AT, in both mice and humans.
Okay, and how are they different from, say, a lymph node?
Well, unlike lymph nodes, FALCs don't have a distinct capsule around them,
Hmm.
and they're in direct contact with the fat cells, the adipocytes.
I see.
Since they were first discovered, they've been found in various VA depots, mesenteric, mediastinal in the chest, gonadal, pericardial, and also in perivascular adipose tissue, PVA, the fat around blood vessels.
Mm.
But interestingly, not really found in subcutaneous fat, SAT.
Okay, so mainly a VAT phenomenon.
Seems that way. And their internal organization is different too. No clear separate zones for B and T cells like in a lymph node. Usually you see dense clusters of B cells, some T cells and other immune cell types mixed in. We see them in PVA too, both normally and in diseases like atherosclerosis.
So the immune landscape in fat is really specialized depending on location.
Absolutely. And just to complete the picture, B cells are also found in brown adipose tissue, B8, in mice. Their numbers seem to go up when mice eat a high-fat diet, but exactly what they're doing there, well, it's still being actively investigated.
It really drives home that fat isn't just storage, is it? It's an active immune organ.
Couldn't have said it better myself. It's incredibly dynamic.
So how do these B cells, particularly in VAT, contribute to the inflammation we often associate with obesity?
That's a crucial question. The research suggests there's an early buildup, an accumulation of B cells in VAT when mice are put on a high-fat diet. And this happens, interestingly, even before we see big changes in other immune cells like T cells or macrophages.
So B cells might be some of the first responders in a way.
It looks like it, yes. And some studies point towards the B2 cells specifically playing a role in promoting inflammation during this HFT-driven fat expansion.
How do they do that?
It seems they contribute by producing inflammatory IgG antibodies and also releasing those pro-inflammatory cytokines we talked about, like TNF-alpha.
Okay, so B2s and IgG lean towards inflammation here. Does this hold true in humans?
Well, there are similarities. Human omental VAT also tends to have higher numbers of B2 cells compared to subcutaneous fat, SAT.
But what about SAT? Is it just quiet immunologically?
Not entirely. There's evidence for local antibody production happening in human SAT too. Researchers have found B cells in SAT making IgG and cytokines. They've even spotted germinal center B cells and follicular helper T cells there.
Germinal centers. Those are like training grounds for B cells to make better antibodies, right?
Exactly. And follicular helper T cells, or TFH cells, are crucial partners for B cells in those germinal centers. Finding them in SAT suggests that sophisticated antibody refinement is happening locally, right there under the skin.
Yeah.
It's not just antibodies made elsewhere drifting in.
Okay, so B cells are active in both VAT and SAT, but maybe different types dominate or have different roles. What are they actually reacting to? What triggers them in the fat tissue?
That's still a bit of a mystery, honestly. A leading idea, though, involves stuff coming from our diet, or rather our gut bacteria responding to diet.
How does that work?
Well, a high-fat diet can sometimes compromise the gut lining, making it sort of leaky. Increased intestinal permeability is the term.
Okay.
And studies in mice show this can let bacterial components like lipopolysaccharide or LPS, part of the outer wall of certain bacteria, leak out of the gut and into the bloodstream or nearby tissues.
And the immune system sees that as a threat.
Exactly, it can trigger inflammation. And indeed, researchers have found higher levels of circulating IgG antibodies that target gut bacterial antigens in both HFT-fed mice and in humans with obesity.
So the B cells in the fat might be reacting to these escaped bacterial bits?
That's a strong possibility. It's also thought that maybe B1 and B2 cells respond differently. They might recognize these antigens, or maybe HFD-induced molecules, using different receptors like toll-like receptors, which sense general microbial patterns, versus their highly specific B-cell receptors. But pinning down the exact antigens and how each B-cell subset responds in AT, yeah, that still needs more work.
Okay, so B2 cells seem potentially pro-inflammatory in obesity. What about the B1 cells, especially those ones concentrated in the omentum?
Here's where it gets really interesting, sort of the flip side of the coin. In contrast to the inflammatory role suggested for B2-derived IgG, the natural IgM antibodies produced mainly by B1 cells seem to be anti-inflammatory.
IgM versus IgG.
Yeah, the sources really highlight that B1 cells can actually reduce VAT inflammation, improve glucose tolerance, and lessen insulin resistance in obese mice.
So they're protective. How?
It's thought to be partly through producing that anti-inflammatory cytokine IL-10 and also through secreting a broad range of IgM antibodies, what we call polyclonal IgM.
Polyclonal meaning they target many different things.
Yes, not super specific like some IgG, but offering broader protection. And even within B1 cells, there are subsets, B1A and B1B. Both seem to chip in with IgM production and dampening inflammation.
Okay.
One study even identified a specific molecule, a transcription factor called ID3, as important for regulating the number of B1B cells and how much IgM they make. Mice engineered to lack ID3 just in their B cells actually had more B1B cells and higher IgM levels.
And what happened when they got fat?
When they were fed a high-fat diet, they showed less inflammation in their VAT. And even more convincingly, if you took those ID3 deficient B1B cells and transferred them into mice lacking B and T cells altogether, those transferred B1B cells homed into the VAT, started secreting IgM locally, and actually improved the recipient mice's glucose tolerance and insulin sensitivity.
Wow, that's pretty direct evidence for a protective role. Does this link hold up in humans?
There are suggestive links.
Yeah.
As mentioned, B1 cells are enriched in human omental VAT, and researchers have found an inverse correlation, meaning when one goes up, the other goes down, between the levels of IgM antibodies targeting certain oxidation-specific molecules, damage markers essentially, and levels of an inflammatory marker called MCP1 in people's blood.
So more of this protective IgM seems to correlate with less inflammation.
That's the idea, yes. It really suggests that maintaining a healthy balance between B1 and B2 activity in our fat tissue could be crucial for metabolic health.
It really paints a complex picture.
Absolutely. And while we've learned a lot, the research also points out, knowledge gaps. We still don't fully know the specific antigens these B-cell antibodies are targeting in AT, and the exact role of those stimulatory signals, the second key for B-cell activation in AT inflammation, isn't totally clear yet.
Still questions to answer.
Always in science. But the findings so far are definitely exciting. They hint at possibilities for future immunotherapies that could perhaps target adipose inflammation, specifically to help with related conditions like heart disease or diabetes.
Really fascinating stuff. Okay, let's make that big shift now. We're moving from fat tissue immunology to the front lines of the fight against cancer, looking at a really innovative therapy approach.
Yes, the second source we looked at explores a treatment strategy combining low-dose chemotherapy with inhibition of immune cells called macrophages. This was studied in a mouse model of triple-negative breast cancer, or TNBC.
TNBC, that's known for being quite aggressive, isn't it?
It is, yes. It can be very challenging to treat effectively. And this study uncovered a really intriguing and potentially critical interaction between B cells and T cells that seemed essential for getting long-term tumor regression.
Okay, B cells and T cells working together against cancer. Before we dive into the results, maybe we should tackle some of the key terms here too. There seem to be quite a few.
Good idea. It'll help frame the findings. So we'll talk about the tumor microenvironment, or TME.
Right, the whole ecosystem around the tumor.
Exactly. Cells, molecules, blood vessels, the works. Within the TME, you often find tumor-associated macrophages, TAMs.
Macrophages again, but these ones are often bad news in cancer.
Often, yes. In many cancers, they can be immunosuppressive, meaning they actually dampen the immune response against the tumor. They can help the cancer hide or grow.
Okay, so we want to get rid of those or change them.
That's often a goal. On the other side, you have cytolytic T cells, also called CD8 plus T cells. These are the killer T cells, the primary soldiers that directly kill cancer cells.
The good guys we want more of.
Right. Immunostimulatory effects just means treatments that boost the immune system's cancer-fighting ability. Now, the study used low-dose chemotherapy, specifically a drug called cyclophosphamide, or CTX.
Low dose. So not the usual high doses meant to wipe out cells?
Correct. Here, the idea is that lower doses might actually modulate the immune system in beneficial ways rather than just causing widespread cell death. It's sometimes called metronomic chemotherapy.
Interesting. And they combine this with macrophage inhibition.
Yes, using a drug called pexidartinib, or PXB. It's a CSF1R inhibitor. CSF1R is a receptor crucial for the survival and function of many macrophages, so blocking it dampens macrophage activity.
Okay, so low-dose chemo plus blocking the potentially bad macrophages.
That's the combo. We mentioned TNBC is aggressive. Metastasis is when cancer spreads to other parts of the body.
Right.
A key technique used was single-cell RNA sequencing, cRNA-seq. This lets scientists look at the gene activity in thousands of individual cells. Really powerful for dissecting complex environments like the TME.
Gives you a very detailed cellular map.
Exactly. We'll also mention T helper cells, or CD4 plus T cells. They're like the coordinators or generals of the immune response, helping activate other cells like B cells and killer T cells.
Okay.
Antigen-presenting cells, APCs, are cells that display antigens like bits of tumor protein to T cells, activating them. B cells can act as APCs too, which is relevant here.
Ah, okay. So B cells can present antigens to T cells.
Yes. Anti-tumor immunity is just the immune system fighting cancer. Long-term responses, or durable regression, means the tumor stays controlled or gone for a long time.
That's the ultimate goal.
Absolutely. High-dimensional imaging, like imaging mass cytometry, IMC, lets researchers visualize many different protein markers on cells within tissue slices simultaneously. It gives spatial context, who is next to whom.
See the cells interacting in their neighborhood.
Precisely. Tertiary lymphoid structures, TLS. These are organized clusters of immune cells, sometimes resembling mini lymph nodes that can form right inside tumors. Their presence is often linked to better outcomes with immunotherapy.
TLS. Okay. Important structures.
You'll see markers like B220 plus CD86 plus a CD40 meal plus other. These are just protein labels on cell surfaces. B220 is common on B cells. CD86 and CD40 feature are markers of activation, often seen when B cells and T helper cells are interacting.
Indicators of activity.
Right. T cell receptor, TCR repertoire, refers to the diversity of TCRs in the body, determining what antigens T cells can recognize. Checkpoint blockade is a major type of immunotherapy, drugs that release the brakes on T cells.
Like PD-1 inhibitors.
Exactly. Pathologic complete response, PCR, often used with neoadjuvant chemotherapy, chemo before surgery, means no detectable cancer cells are left in the surgically removed tissue.
A very good sign.
A very good sign indeed. We mentioned CSF1R inhibitor. Ki67 plus marks cells that are actively dividing. F480 plus is a common mouse macrophage marker. CD127 plus is often on memory T cells.
Lots of mark.
There are. Others like SMAD7 plus EN and Favios Lysix both get 1, GZMBI67 plus SAY, LAGA3 plus mark, different T cell states, naive, memory, effector, killer, proliferating, exhausted. UAP is a way to visualize complex single-cell data. CCR7 plus 7, CD69 plus SAY, FOXD3 plus mark, different CD4 plus T cell types, naive, activated, regulatory. An activated B cell, ABC signature, is a pattern of gene expression seen in activated B cells. Adriamycin is another chemo drug, and TILs are tumor-infiltrating lymphocytes, any immune cells within the tumor.
Phew. That was a list.
Okay.
But super helpful to have those defined as we go. So what happened when they used this low-dose CTX plus PXB combo in the TNBC mouse models?
The results were quite striking. This combination therapy proved remarkably effective. It successfully treated aggressive mammary tumors and significantly reduced or even prevented lung metastasis in several different mouse models of TNBC.
Wow. Successful treatment even with aggressive tumors and metastasis. That is promising. And what was the B-cell connection? Where did they come in?
This is where it got really interesting. Using that detailed scRNA-seq and the high-dimensional imaging, the researchers discovered this crucial coordinated interaction between T helper cells, the CD4 plus ones, and antigen-presenting B cells.
The B cells presenting antigens to the T helper cells.
Exactly. And this interaction seemed absolutely fundamental for driving a strong and, importantly, sustained anti-tumor immune response that led to those long-term remissions they saw.
How did they see this interaction happening?
With the imaging, they could literally see activated B cells marked with B220 and CD86 nestled right up close to activated T helper cells, marked with CD40 helperands and CD4. And these cozy pairs were found within those tertiary lymphoid structures, the TLS, even weeks after the treatment had finished.
So these activated B cells and T helper cells were hanging out together in these immune hubs within the tumor long after the drugs were gone.
That sounds like immunological memory being established.
That's exactly what it suggests. It implies a lasting change in the tumor microenvironment driven by this B cell-T cell partnership. Okay, that's cool in mice, but doesn't mean anything for human patients.
That's the crucial next step they took. They looked at data from human TNBC patients who had received standard neoadjuvant chemotherapy, and they found that specific B-cell markers and gene expression patterns, signatures derived from those expanded activated B cells they saw in the successfully treated mice, actually correlated significantly with achieving a pathologic complete response, PCR, in the human patients.
Wait, so the same B-cell signatures that marked success in the mice also predicted complete response in humans?
Yes. Particularly, an activated B-cell cluster-derived signature they identified showed a strong association with PCR. They also found that simply having more B cells in the tumor tissue overall, measured by markers like CD19 and CD20, was linked to complete responses in the human patients.
This is really exciting. It bridges the gap from the mouse model directly to potential clinical relevance.
It absolutely does. This research strongly suggests a potential new way forward for TNBC treatment. The idea is that by carefully combining low-dose chemo with macrophage inhibition, you might be able to specifically foster this beneficial, long-lasting B-cell-mediated T-cell immunity within the tumor.
Create an environment where the immune system can learn to fight the cancer more effectively and durably.
Precisely. It's about harnessing the immune system's own power through understanding these intricate cellular collaborations.
Truly groundbreaking work, highlighting how understanding these immune interactions could lead to totally new therapeutic strategies. Okay, one more fascinating piece of research for today. Shifting focus again, this time to asthma.
Right. Our third source zooms in on the interplay between a specific type of macrophage, the M2 macrophage, and B cells, specifically in the context of asthma. It shows that these M2 macrophages can actually encourage the production of regulatory B cells, BREGs, or IL-10 plus B cells, which in turn can help alleviate asthma symptoms in mice.
Okay, M2 macrophages, helping B cells become regulators to calm down asthma. Let's quickly define the key terms for this section too.
Sure. Asthma, most people know, is a chronic respiratory disease, inflammation and narrowing of the airways, causing wheezing, coughing, shortness of breath.
Right.
Macrophages, we've met them twice now. Here, the key distinction is M1 versus M2. M1s are generally pro-inflammatory, fighters. M2s are more anti-inflammatory, involved in tissue repair, immune regulation. Researchers often use specific signals to push macrophages toward one state or the other in the lab, like LPS and IFN gamma for M1, or IL-4 and IL-13 for M2.
So M2s are the calm down and repair type.
Broadly speaking, yes. Regulatory B cells, or BREGs, are a B cell subset whose defining feature is producing the anti-inflammatory cytokine IL-10. Their job is to suppress excessive immune responses, keep things in balance.
IL-10 B cells. Okay.
TGF-beta, transforming growth factor beta, is another important regulatory cytokine. It has many roles, often involved in suppressing inflammation. Oxidative phosphorylation is a core metabolic process cells use to generate energy, ATP.
Like the cell's power plant.
Pretty much. RNA sequencing, RNA-seq, again looks at gene expression. KEGG and GSEA are computational tools to find biological pathways that are active or changed based on that RNA-seq data.
Analyzing the gene patterns.
Correct. A transwell culture system lets you grow two cell types in the same dish, but separated by a filter with tiny pores. They can share soluble factors, like cytokines, but can't touch directly.
Useful for figuring out if signaling is contact-dependent or through secreted molecules.
Exactly. Luminex and ELISA are lab techniques to measure the amounts of specific proteins, like cytokines, in samples. And anti-CD40 stimulation is just a way to activate B cells in a lab dish by triggering their CD40 receptor.
Got it. Okay, so in their asthma mouse models, what did they find about macrophages initially?
Well, first they did an experiment where they depleted macrophages overall in the asthma model mice. And interestingly, getting rid of macrophages actually made the asthma symptoms worse.
Oh, so macrophages overall seemed somewhat protective.
It suggested that yes. And importantly, this worsening of symptoms coincided with a reduction in those beneficial IL-10 producing regulatory B cells.
Which hints that maybe macrophages are needed to support these regulatory B cells.
Exactly the line of thinking. So they investigated further, and the key finding emerged. It was specifically the M2 polarized macrophages that promoted the development and function of these IL-10 BREGs.
How do they show that?
They co-cultured M2 macrophages with B cells in the lab. When they did this, they saw a significant jump in IL-10 production by the B cells. These B cells essentially became regulatory B cells.
And did these educated B cells actually work? Could they regulate?
Yes, they tested them and found these M2 educated B cells had negative regulatory abilities. They could suppress the proliferation and effector functions of T cells, which are key drivers of asthma inflammation.
And if they used M1 macrophages instead?
No effect. Co-culturing with M1s didn't induce the IL-10 brags, so it's specific to the M2 interaction.
Okay, so the M2 calming macrophages help create the IL-10 calming B cells in asthma. How does that communication happen? Is it direct contact or signals?
They looked into that mechanism.
Oh.
RNA sequencing suggested it was likely cytokine-mediated. To test this, they used that transwell system, separating the M2s and B cells with a filter. And even without direct contact, they still saw the induction of IL-10 plus B cells. That strongly pointed toward soluble factors, cytokines being the messengers.
Okay, so which cytokine was it?
They measured various cytokines produced by the M2 macrophages and zeroed in on TGF-beta1. It seemed to be the crucial driver.
How did they confirm it was TGF-beta1?
They did blocking experiments. They added an antibody that specifically neutralizes TGF-beta1 to the co-culture. And when they did that, the ability of the M2 macrophages to induce IL-10 and B cells was blocked. That really cemented TGF-beta1 as the key signal.
Makes sense. Any other clues from the RNA sequencing?
There were hints that metabolic pathways, specifically oxidative phosphorylation, might also be involved in how the M2 macrophages support this brag activity, but that needs more investigation to fully flesh out.
So the main story is M2 macrophages release TGF-beta1, which tells B cells, hey, become IL-10 producing regulators.
That's the core mechanism they uncovered yet.
And the implications.
Functionally, it means these M2-educated brags can put the brakes on the overactive T-cell responses seen in asthma. Therapeutically, this is quite exciting.
How so?
It suggests that targeting this specific M2 macrophage B-cell interaction to boost the production or function of these regulatory B cells could be a completely new way to treat asthma. Instead of just broadly suppressing inflammation, you'd be enhancing a natural regulatory pathway.
A more targeted, perhaps more nuanced approach.
Potentially, yes. It's another example of how understanding these specific immune cell dialogues opens up new therapeutic possibilities.
That's a fantastic insight, rounding out our exploration today. Thank you so much for guiding us through these really diverse roles of B cells.
It's been my pleasure. They really are fascinating cells, aren't they? Involved in so much more than people might think.
Absolutely. So we've journeyed through B cells in the inflammatory landscape of fat tissue, their unexpected but crucial role in a novel cancer therapy, and their regulatory function in asthma via interactions with M2 macrophages. It's abundantly clear that B cells are far more versatile than just being antibody factories. Their interactions within different tissues have really profound impacts on our health and on disease processes.
Definitely. And the research is constantly revealing new facets, new jobs they have, new ways they interact.
Which offers, as we've seen, really exciting possibilities for future treatments and interventions. We hope this deep dive has sparked your curiosity and given you some valuable insights into the world of B lymphocytes. Thank you for joining us.